I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, everyone. My name is Grant Holly. I'm the Executive Director of the Wisconsin State Cranberry Growers Association. We are very happy to be able to partner with UW Extension to bring you the research needed to be able to navigate our ever-changing environment. I'd like to thank all of our speakers today for their valuable time and leadership, the growers for joining us, and Allison for being the catalyst from which all of these sessions rely. Thank you so much.
And want to give a big thank you to Sandeep and to Cesar and to Amaya, who'll be joining us as well, for being willing to present with us in this, our November kind of nationwide roundup virtual brown bag lunch. As in case there's anyone joining us for the first time, we do have these every month that isn't harvest and that Wisconsin doesn't have an in-person outreach event. So we're glad to get to take specifically the November and December sessions to catch up on new research that's gone on through the year, especially things that have happened outside of the state of Wisconsin, and make sure that everyone is up to date on the cool new things that are happening. To that end, today we're going to have Cesar Rodriguez-Saona from Rutgers is going to present some new kind of up-to-date information on the Blunt Nosed Leafhopper. They've been doing more work in the past couple of years and going to make sure that we have everything as up-to-date as possible on that. Sandeep Bhatti is at UMass and is a postdoc research associate there, and he's been doing some pretty exciting work on frost modeling and has some spring data to share with us. And then our Amaya Atucha of University of Wisconsin-Madison is going to present in the topics of color, especially just before harvest and even after fruit has come off the vine. A couple of housekeeping notes. We have closed captions on. If you prefer to see those, you can choose ... and view closed captions. I have the chat on. We really love to have people ask questions over the course of the event. So if you have a question, you can either unmute yourself and ask it, or you can ask in the chat. You can also use to Allison and ask a question directly to me. And if you do that, I will ask that question anonymously on your behalf. And with that housekeeping out of the way, I think we are good to see if we've got a representative from the Wisconsin State Cranberry Growers Association to give us a welcome, but I don't, if you are here, anyone, go ahead and unmute yourself and say welcome, but I just want to thank, I am the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension, and I get a lot of guidance from the Professional Development Committee of the Wisconsin State Growers Association in terms of getting suggestions for who should speak at these. So always appreciate feedback and make sure we're talking about the most relevant things. So speaking of those relevant things, I am ready to hand control over to Cesar. I will stop sharing my screen here and let you hop on. And as that screen is showing, just thanks everybody for joining. We've got a pretty good crowd for the day before hunting season. And I'll say that's because we
So thank you, Allison, for the invitation. So I'll be talking about our work on Blunt Nosed Leafhoppers in cranberries. So for those of you that are not familiar with this insect, Blunt Nosed Leafhoppers are native to the U.S., the U.S. and also Canada, and they are distributed in the eastern U.S. Where cranberries are grown. It can feed on several hosts on ericaceous plants, so cranberries and related plants, and it has a single generation per year. The eggs overwinter, and then they hatch in New Jersey. They hatch in early May. We take the water off in mid-April, and as soon as the plants start to put new growth, that's when the eggs start to hatch. It takes the nymphs about a month to reach adulthood. So we start to see adults at the end of June through July. And the eggs are laid, obviously, when the adults are present throughout July and beginning of August. So this is a problem because it's a vector of a phytoplasma that causes false blossom disease. So this disease is characterized by these malformed flowers, as you can see in the picture. It causes the flowers to stand erect, and also it causes the petals to become redder than normal. It also has other symptoms like witch's broom, and so I said that. So this is a drawing that we recently did on the life cycle of the insect. As I mentioned, the first instars, the eggs start to hatch in May, so we start to see the first instars by mid-May, early May, mid-May. We start to see the second instars by the end of May and beginning of June. Also, there's some overlap among instars, so we start to see also the third instars towards the end of May, beginning of June. The fourth instars in June by mid-June. The fifth instars towards the mid-June through the end of June, early July. And as I mentioned, the adults start to emerge towards the end of June and through July and the beginning of August. And that's when the eggs are laid. I want to share a video that we created this year that provides information about this insect and the disease that they transmit.
The American cranberry is a native crop which has been cultivated in the U.S. for a little over 200 years, showing up on Thanksgiving tables across the country. 2023 alone, upwards of 8 million barrels of cranberries were produced in the United States, with a production value of nearly $35 million. However, with the increased restrictions on pesticide usage since the early 2000s due to environmental safety reasons, cranberry bogs are now seeing the resurgence of one of their most dangerous pests, the blunt nosed leafhopper. The blunt nosed leafhopper is a piercing, sucking insect that feeds on sap from the stems of cranberry plants. The main concern regarding this leafhopper is not the damage done by the insect itself, but rather the phytoplasma it can transmit, which causes cranberry false blossom disease. The blunt nosed leafhopper has 7 life stages overall. It completes 1 generation a year and overwinters as an egg. They have 5 sexually immature stages over the course of 1 to 2 months before becoming adults and laying eggs. The adult leafhopper is easily distinguishable from the instars by its size and presence of wings. The lack of wings, and therefore flight in immature stages, means that if they are found in a bog, the eggs were likely laid there the year before. The adults appear by the end of June, with males emerging a few days before the females. They're most abundant in July, with their numbers starting to diminish by the end of that month. The winged adults have a characteristic blunt head and vary from light yellowish gray to a dark brown. To monitor for blunt-nosed leafhopper, you need to sweep your field to see if you catch the insect or not. In New Jersey, it's recommended to start sweeping your field mid-May, which is around 3 weeks after they drain the bogs, and then almost weekly until bloom to make sure that you know your numbers of blunt-nosed leafhopper. To sweep for the blunt-nosed leafhopper, you take your sweep net and using 180 degrees, you sweep it through the bog. 25 sweeps consist of 1 sweep set, and it is generally recommended to do 1 sweep set per acre. If you can't do that, then 1 sweep set or 2 sweep sets per bug also will do. Then after you sweep, you close the net so that none of the hoppers escape. You take your rusty, dusty plastic bag and empty out the contents from the net into the bag. After you did this, you could continue sweeping the rest of the field. And it's important also when you sweep your bug to sweep at different sections of the bug, because the insects can aggregate in one section over another. So to actually make sure you get a whole read on your presence of the leafhopper, it's important to sweep in different parts of your bug. We have this sample that we collected from earlier, and you see it has all of these little cranberry leaves all over the place. So we gotta filter it out. Get a couple filters for the sample. So this is a lot of the leaves that we just took out. And we are left with here a lot of little specks. All right, so when we look here under the microscope, we could see all sorts of different things. So there are, first off, just dirt and rock particles, but there could be other insects, such as whiteflies or columbella, maybe some thrips can be in the area, and some mites. What we are looking for are blunt nosed leafhopper. This is here a young instar. Essentially, you have to sort through this to make sure you find the ones that actually are important for this. Cranberry false blossom disease is characterized by a malformation of the flowers. Symptoms include erect, misshapen flowers that do not produce fruit, leading to devastating effects on cranberry yields. Diseased plants grow their uprights close together, forming a witch's broom, and then turn reddish earlier in the fall than healthy plants. While the blunt-nosed leafhopper is not the only type of leafhopper out there, it is the only one known to spread cranberry false blossom.
So hopefully you found that information useful. So we tried different methods to improve our monitoring techniques for a blunt-nosed leafhopper. One of the things that we tried in the past was to try to capture the adults using sticky traps. However, for this we tested like 5 different colors. So we tested yellow, red, green, white, and blue, and we compared that to a clear sticky card. And what we found was a sharp-nosed leafhopper was a clear preference for yellow cards, followed by red and green. However, blunt-nosed leafhopper did not have a clear preference for these colors. They had a higher tendency that was significant to be captured in red and green traps compared to the other colors. But the numbers were not that high. So this is what we currently recommend our growers to use for controlling leafhoppers. Pyrethroids work. We know that organophosphates also are effective in controlling leafhoppers like diacenon. Also, we found that 7 carbamate has a good efficacy. Right now, our growers are relying mainly on the use of fanfare pyrethroid for the control of these pests. However, you have to be careful when you're using an insecticide repeatedly that has the same mode of action. So we recommend growers to rotate insecticides that have different mode of actions to avoid the onset of resistant populations. So for that purpose, we're looking at a new insecticide. So in recent years, we have been testing a new product from Syngenta. We compare that with a grower standard, such as the pyrethroid. So I'm just going to show you like one set of data that we have However, the results are very consistent. So this is trials where we spray, we apply these insecticides to foliage in the field, and then we bring the foliage to the lab. We place them in containers, and we release nymphs inside these containers, and then look at mortality 1, 3, and 7 days after exposure. So as you can see here, so this is the unregistered insecticide on the left, a danidol in the middle, and the control on the right. And you see the blue is the 1 day after exposure, the orange 3 days, and the gray 7 days after exposure. And what you can see is that the unregistered insecticide doesn't kill the nymphs right away, but it takes some time for them. They need to be exposed for a few days until we see high mortality, compared to an insecticide such as danitol that kills them right away. So we're also conducting studies to better understand interactions between cranberries, the phytoplasma that causes false blossom, and the vector, the leafhoppers, and non-vector herbivores. So in these studies, for example, we have done single choice experiments where we cage nymphs on infected and non-infected plants, and we see no effect on nymphal weights. However, we see that adults are bigger when they feed on infected plants. So there's some kind of benefit of feeding on these plants to the adults. We also, but however, we don't see any effects on survival. So we see an effect on the weight, and survival was the same. When we looked at choice experiments, these are done in olfactometers. So we're giving, in this case, a choice to the nymphs between healthy plants and air. We see that the nymphs prefer to go to the choice that has the healthy plants. So they are attracted to the smell of healthy plants. However, they were not attracted to the smell of false blossom infected plants. And there was no difference when we gave them a choice between healthy and infected plants. The adults, we see the same pattern. Adults are attracted to healthy plants, so they can smell the healthy plants and be attracted to those odors. However, this does not happen when we expose them to false blossom infected plants. And also there was no difference between healthy and false blossom infected plants. And what we find is that the healthy plants emitted higher quantities of volatiles that are likely attractive to these leafhoppers. We also see that infected plants make the non-vectors, herbivores, bigger. So there's, when we test a spotted fireworm, a spaghetti fruitworm, and a spongy moth, they grow bigger when they feed on infected plants than uninfected plants. And also they ate more of the leaves. And this is due mainly because there's higher amount of nutrients in infected plants, especially nitrogen levels are higher in the infected plants than non-infected plants. And also we see a reduction in the number of defenses, the amount of defenses, for example, proanthocyanins that can have an effect on herbivore growth. So we see that infected plants have lower quantities of these defenses compared to uninfected plants. So what are some management guidelines that we recommend our growers? First is to identify infested areas. So rely, as we show in the video, you have to monitor the leafhoppers using sweep net before bloom to collect the nymphs. And you can freeze them and then count them under a microscope. We perform sets of 25 sweeps. We recommend one sweep per 1 to 10 acres, at least 10 sweeps per 10 to 20 acres, or one sweep per 2 acres larger than 20 acres. So currently there's a lack of an economic threshold, and this developing an economic threshold for an insect that causes that transmit a disease can be challenging because it depends on the proportion of individuals that are infected with the phytoplasma. And also the populations can vary by variety and location. So what we have noted, though, is that higher infection rates have been observed in newer varieties, even when populations of leafhoppers is low. So right now, what we are recommending as a control measure for blunt nosed leafhoppers is to look at your history of insecticide applications, and also depending on the cranberry variety that you grow, we recommend growers, if populations are high, that to treat using pyrethroids, carbamates, or organophosphates. There's some other classes of insecticides that seem to be effective, like sulfoxamines, like Closer, and neonicotinoids also are effective. However, neonicotinoids we don't tend to recommend before bloom because potential effects on pollinators. And Closer, we don't have enough data at a field level effect, at a field level to recommend to our growers yet. So that's all I have. I don't know if we have time for questions or.
I think we're running just exactly on time, so I think the best thing will be to have all the questions for all the presenters at the end of the hour, if you're able to do that. We've got that 5 minutes set aside for questions. That was fantastic. Have gained a lot of information on blunt nosed leafhoppers since the last time we've had an update. So I appreciate that. Appreciate your students who helped with the research, and thank you so much for being willing to share with us. So I want to give a huge thank you to Cesar and to Sandeep and to Amaya. Thank you all so much for joining us today.
Thank you.
Thank you.
And a lot of the growers are putting a thank you in the chat as well.